to give an overview on the application of some of the genomic insights that we've developed in terms of their application to blood cancer in particular, and how this might lend itself to the notion of personalised medicine. Some of this is relatively old news in terms of it's only 10, 15 years old, but the pace of acquisition of knowledge and change makes it, if something's found 10 years ago, it's quite old, but a lot of it's very, very new. So the talk on personalised medicine and blood cancer is really incomplete without discussing what we call the CML imatinib paradigm. But before we go into that, I'm sure most of you may have heard of imatinib. I thought it was worth having a discussion about tyrosine kinase and some cell biology. So tyrosine kinase is a critical element of cells, particularly cancer cells. We know that activation of tyrosine kinases results in what we call auto or transphosphorylation of tyrosine residues in the activation loop of the kinase. What this simply does, it changes the shape of the kinase, increases the catalytic activity, and generates binding sites for signaling proteins that mediate signaling messages downstream in the cell to modulate cellular processes such as growth, apoptosis, and proliferation. So very simply, how these kinases can become activated in cancers and blood cancers, you have a transmembrane receptor tyrosine kinase here that upon binding of ligand can dimerize. It has a conformational change that makes it phosphorylated and bind effector proteins that then pass those phosphate residues on and activate downstream signaling proteins. In cancer, these can become activated via a number of mechanisms. One can be the overexpression of the receptor tyrosine kinase, so you simply have either an amplification or an overexpression of the gene, you have more of them. You can also have increase in the amounts of ligand around that are stimulating those receptors, or you can have a combination of the two. Furthermore, and this is of relevance to leukemia, we can have mutations that Paul mentioned in his talk on a gene called FLT3, where internal tandem duplication, so repeated abnormality within the gene, causes a conformational change whereby this no longer needs to bind the ligand, although it still can, and it has a conformational change that turns it on all the time. It's constitutively active. And so that's sending a proliferative signal to the cell pretty much all the time. And finally, you can have gene fusion events. So in the case of CML, which I've mentioned, we have what we call the Philadelphia chromosome, or the 922 translocation, which brings two genes together, BCR and ABL, which results in a conformational change to the ABL1 kinase, and again, constitutive activation and stimulation of downstream signaling pathways. So these are the sort of hallmark events that result in kinase activation in cancer. So to get back to imatinib, so a small molecule that targets this activated ABL kinase as a result of the Philadelphia chromosome 922. This has been known for many years, and the reason why is because we can detect this with conventional karyotyping and cytogenetics. It can be inhibited with these small molecules, imatinib, dasatinib, nilotinib, ponatinib. Not surprisingly, when one drug is spectacularly effective, then there's a search for more potent and more targeted agents. It basically inhibits ATP binding into the binding pocket, and therefore prevents phosphorylation, so transferring the phosphate residue from ATP down onto the tyrosine residues, and prevents the activation of downstream pathways. Importantly for CML, it's really revolutionised this disease, and we now refer to what's called the pre-imatinib era, where this disease was only curable by hematopoietic stem cell transplantation or bone marrow transplantation. It's a remarkably well-tolerated drug. When I say that some of this is old, it's only 14 years ago that this plenary paper was published in the New England Journal by Brian Drucker, a phase 1 study in patients with CML, a single oral agent taken daily, resulted in 53 of 54 patients having a complete hematological response, and remarkably it occurred within the first 4 weeks of treatment in most patients. Not surprisingly, from an academic perspective, this paper's got some 4,500 citations, and that number continues to rise. This really set the scene for targeted therapy, certainly in leukemia, but arguably in cancer in general, and hence why we refer to it as the imatinib paradigm. Importantly, in childhood acute lymphoblastic leukemia, the presence of the BCR-ABL from the Philadelphia chromosome is always been a bad thing to have, and by childhood ALL standards it still is. Again, this disease, even with bone marrow transplantation, was only curable in less than a third of patients, but with the addition of imatinib to intensive chemotherapy, you don't need to be a rocket scientist to see that the Kaplan-Meier curve here is dramatically improved, and almost 80% of these patients can be cured. And more importantly, long-term follow-up of these patients published last year shows that we don't need bone marrow transplantation to cure these children. We can cure them, at least 75% of them, with imatinib alone. And whether they get a transplant or not, if they've had imatinib, their survival is excellent. So this has really changed the outcome, certainly for Ph-positive ALL. As I said, this was all based on conventional karyotyping. We can see the 922 translocation by FISH and by conventional karyotyping. However, we know that there are a large proportion of patients that don't have didn't have an identified genetic lesion when it came to ALL. And in recent years, this has spawned a brand new subset of childhood ALL, and indeed it's identifiable in adult ALL and arguably more commonly in adolescents and young adults than children, in fact. And it's called PH-like ALL. It's called PH-like ALL because it has a very similar gene expression profile to ALL and has very similar cellular effects. Many groups both in Europe and the United States found that the gene expression profile in PH-like ALL was almost indistinguishable from patients with PH-positive ALL. Importantly, these lesions are targetable, and we were very fortunate to contribute to a landmark study by Charles Mulligan and colleagues last year that showed that these lesions could be targeted with small molecule inhibitors such as imatinib for patients with this new disease entity called PH-like ALL. Importantly, this subset of ALL collectively confers a poor prognosis. These patients have higher relapse rates, higher rates of induction failure, and inferior survival. And using a combination of RNA sequencing, whole exome sequencing, whole genome sequencing, we're able to identify a whole myriad of mutations that were completely unknown prior to the application of new genomic technologies. And these can be grouped into different classes of events. Many of them will have what we call an ABL class fusion, so a gene fusion involving the ABL1 gene itself or involving other kinases that cause similar intracellular signaling cascades. And again, these can be inhibited by imatinib or dasatinib, for example. Other mutations here, such as mutations involving JAK2 or CRLF2 overexpression in gene fusions, cause activated JAK-STAT signaling. And again, these are targetable. I'll show you how in a moment. And then there's a myriad of other mutations that affect other intracellular pathways, RAS pathway mutations. These can be targeted with small molecule inhibitors of kinases like MEK kinase, for example. So this has really caused an explosion of knowledge in this and a generation of a new subtype of ALL, which we knew nothing of before. And the promise is that these are all therapeutically targetable. As you can see from cellular biology, this is real and causes real biological effects. When you transfect in these genetic events into cytokine-dependent mouse cell lines, they are no longer cytokine dependent. They just grow happily without being stimulated. So that tells us that these are activating mutations that stimulate the cell to grow. You can inhibit the growth of these cells with targeted small molecules, but most importantly, you can inhibit them with targeted therapies based on the target that's activated. So to walk you through this slide here, it's quite important. If you have an ABL1 fusion and mutation, you've got activated phospho-STAT5. So this is a key downstream signal that's turned on in these leukemias. But the signal is only turned off by dasatinib if you've got an ABL fusion. If you treat it with, here in green, if you treat it with ruxolitinib, which targets JAK2, it has no effect. And this is what's shown up here in this cell growth assay. Similarly here, mutations involving CSF1 and SBP2 are inhibited by dasatinib. Dasatinib here shifts the phospho signal back to the baseline, but ruxolitinib doesn't. And similarly, if you've got an activation of JAK2, the JAK2 inhibitor will inhibit the phosphorylation of STAT5, whilst dasatinib, an inhibitor of ABL, has no effect. So this is really pairing the targeted treatment to the mutation that's stimulating the cell. So that's acute lymphoblastic leukemia, highly common in children, less common in adults, but as I've said, pH-like ALL is certainly more common in adults than it is in children, as is pH-positive ALL. So to acute myeloid leukemia, FLT3 is an important gene, both prognostically and as a bona fide therapeutic target. And Paul mentioned this briefly in his talk prior. We know that internal tandem duplication of the FLT3 gene is prognostically important. These patients, again, have a high risk of relapse and inferior overall survival, particularly when they have a high allelic ratio, so a high mutational burden of FLT3. In particular, you can do this by having both copies of your FLT3 being mutated, or you can lose the wild-type allele. And this is common for both adults and children. Go back to the slide. This is the gene. The mutation causes that conformational change to turn it on all the time. So importantly, the Royal We have been targeting FLT3 for well over a decade now, and we know that we can induce remissions in patients, although they're quite transient, and it hasn't really been known why that is the case. A landmark paper in Nature a few years ago really showed that ITD is a valid target and FLT3 is a valid target in AML in patients with highly refractory disease, relapse refractory AML, and importantly, FLT3 mutated AML. We're pairing our targets to our drugs. We had a 45% response rate. Of some concern though, 8 patients relapsed, and using next generation sequencing technologies, such as the PacBio, which we have upstairs, as Paul mentioned, all 8 cases of patients who relapsed after achieving a complete remission with this potent FLT3 inhibitor, AC220, all had a secondary point mutation in that FLT3 gene. So they had the FLT3 ITD, but the cells are what we call addicted or onco-addicted to FLT3. They like to have FLT3 turned on. If you turn it off, the leukemia cell wants to try and switch it back on. So it's, in this setting, has introduced a point mutation in the kinase domain, which has altered the shape of it and stopped the small molecule binding to it, and has therefore overcome that resistance. During my PhD, we made a cell line model that recapitulated the human disease very nicely, further validating that this is a biological process of importance. So the challenge from a drug development point of view is how do we make small molecule inhibitors that can target not just FLT3 ITD, but also the secondary mutations and therefore prevent the emergence of relapse and resistance. In terms of AML genomics, AML, as you may know, is the most common acute leukemia in adults. Arguably, if we all live long enough, we will probably all get AML. And it's a large proportion of AML. It is a bit grim, but that may well be the case. 100, you'll probably, well, if you live that long, you probably won't get AML. In terms of AML, we've got a large proportion of patients have previously, until a few years ago, unknown molecular events and unknown mutations that are driving their disease. And certainly by conventional karyotyping have what we call cytogenetically normal AML. So we've been really in the dark somewhat as to what are the drivers of AML, particularly in adults. In the Cancer Genome Atlas work, which was published a couple of years ago in the New England Journal, again, recurring theme in my talk, you may notice, a lot of the landmark papers are all in the New England Journal, and that's with good reason. Sequencing 150 AML genomes by whole exome sequencing and 50 genomes by whole genome sequencing. We've uncovered a myriad of genetic events and molecular abnormalities in these AML cases, and they're recurring. Some of them we've known for a while, such as PML-RARA. This can be targeted, NPM1. But there's a new class of gene mutations in what we call the DNA methylation genes, which cause hypermethylation of typically tumor suppressor genes, which confers a survival advantage to cells. Now, when this work came out and the same group had published on this mutation called DNMT3A, it's very common, say around 25-30% of patients, of adult patients will have DNMT3A mutations, is generally thought to be a poor prognostic marker, but it's unknown as to what's going on with its biology. And another thing to say with the application of genome sequencing, we've uncovered knowledge that we didn't have before on the clonal heterogeneity and the clonal evolution of leukemia. So we know that many patients have AML, but they have multiple subclones, and there's almost a Darwinian selection pressure that is applied to the evolution of those clones within your leukemia, and it's sort of a survival of the fittest. But we see DNMT3A mutations in a number of clones, raising the question, is that a founding mutation, or is it the mutation that's really driving it? In recent work, literally a few months ago, published in Blood by Ben Ebert's group in Boston, has shown that these DNMT3A mutations are actually present in our normal hematopoietic stem cells. So, several of us may be harbouring a mutation in our hematopoietic stem cells of DNMT3A, and these persist over time without causing a phenotype like myelodysplastic syndrome or AML, and you need to acquire additional mutations over time to transform it. And this has led to literally a brand new disease, or it's not a disease entity as such, but a brand new classification, which they've coined as CHIP, or clonal hematopoiesis of indeterminate potential. And this is analogous to MGUS in multiple myeloma. But the importance of this is that this is the schematic that's hypothesized to be occurring. As these are in your normal hematopoietic stem cells, over time you acquire mutations in genes like TET2, DNMT3A, so methylation genes, and then over time you acquire secondary mutations that may transform you into MDS, and then additional mutations. Again, you can see this Darwinian clonal branching pattern. Different cells will acquire mutations, or the same cell will acquire a mutation, then it'll acquire a secondary mutation to tip you over into AML. And what's hoped is that with understanding this process further, which is only possible through the application of genomics, the application of hypomethylating agents, for example, there's likely to be other drugs that may do this as well, such as azacitidine, decitabine, may help push patients back from MDS into CHIP and potentially from AML back to MDS and reverse this process somewhat. As I said, this is all very much hot off the press, was a source of much discussion at an AML meeting I was at last week in Budapest, but it's very much an example of how genomic medicine has revolutionised our understanding of leukemia, particularly myeloid leukemias. So in terms of the clinical application, I thought I would also highlight where this is going nationally in paediatrics. I'm a paediatric oncologist. Colleagues of mine in Sydney Children's Hospital and the Children's Cancer Institute at University of New South Wales have launched just 2 weeks ago their Zero Childhood Cancer program. It's a very ambitious program, but it serves to highlight what is actually going on. The plans here are that of children with high risk or relapse refractory cancer will be put through the ringer in terms of genome profiling, drug sequencing, in vivo modelling as well of their disease, all the while conventional therapy or salvage therapy is started, with the expectation that they will have a targetable lesion identified in their tumour that can then refine their treatment, further biopsies and sampling taken, further testing going on, and treatments refined further to hopefully improve outcomes. This isn't pie in the sky, it is ambitious, but it's very much real and it's very much planned to go national. 12 patients will be enrolled in the program next year at Sydney Children's Hospital, with a further 120 patients nationwide enrolled onto that program. With our interest in acute myeloid leukemia in children here at Diamantina and in the genomics resources we have available, the plan is for us to take a lead on the AML aspects of this program, and then eventually by 2020 this should be standard of care, hopefully, in terms of identifying a targeted therapy, almost certainly in combination with conventional chemotherapy granted, to improve outcomes for children with cancer. And this is something that is certainly applicable to adult cancer. So I think this is an indication of where the application of clinical genomics will go, not just in leukemia but in cancer as well. Thank you.